Okay, it's 12:01, so I'm going to get started now. So good afternoon, my name is Francis. I'm a senior lecturer in Public Health Microbiology at Cranfield Water Science Institute at Cranfield University. Lucky enough to be the coordinator of an EBNet sponsored and National Biofilm Innovation Centre supported series, webinar series called the Water Biofilm series. And we've had a couple of seminars so far, and I'd just like to acknowledge the sponsors of both the NBIC and EBNet. So that's UKRI through the BBSRC and EPSRC. And the aim of this series was to establish a series of webinars to look into interesting aspects of water biofilms, focused principally on engineered systems. There's a couple of other groups which have been set up through EBNet to explore more natural biofilm systems and also sensors and other aspects of wastewater treatment. And really, we sit within this area of the National Biofilm Innovation Centre remit. So this is the engineer aspect of biofilms. And there's a number of different themes in which we're going to explore through this series. So this includes wastewater conveyance and drinking water distribution and storage, and that will be the topic of today's talk. And there's a number of challenges, I guess, that we're facing within this within these systems. So could be excessive biofilm growth in sewers or drinking water distribution systems, corrosion, which is increasingly important as we're going into a warming climate with aging infrastructure, water quality concerns, leakage, which has been sort of top of the press at the moment with respect to wastage of potable water supplies during periods of water scarcity, and possibly even innovations such as in-pipe drinking water treatment, for example, using UV LEDs. Another theme is treatment. So this is about how we improve our treatment performance with the same or lower levels of energy, and about managing things like emissions. So greenhouse gases, including nitrous oxide, is top of the agenda at the moment, and how we deal with things like emerging contaminants. So this includes viruses, pathogens, plastics, forever chemicals, and so how we can encourage our biofilms within these systems to do more for us is an interesting area. And then finally, I'm working with a colleague at Cranfield to look into a series of talks on energy and nutrient recovery. So this is through principally wastewater treatment, but getting to ultra low nutrient consents, how we can reach net zero as an industry, and also manage important aspects such as energy security and waste management. So a couple of months ago, we had the first of this series, which was Dr. Henry Lee, who's at Imperial College London, who talked about biofilms within GAC systems for treatment of wastewater, which was a really nice seminar he gave. And today I'm lucky enough to introduce Professor Boxall. Just as a reminder, could delegates who are attending the seminar series please turn your camera and mic off during the actual presentation. Do feel free to ask questions in the chat as you go along so that you can remember them, and then we'll ask Professor Boxall to deal with them at the end through a sort of 10-minute discussion at the end. And then the final point from me is just a reminder, if you are interested in speaking in the water biofilm series, please do get in touch at ebnet@ebnet.ac.uk, or you can email me directly, and my email is at the bottom of the page there. So without further ado, it's my pleasure to introduce Professor Joby Boxall from the University of Sheffield. Joby trained as a civil engineer and did a lot of his early work on mixing and transport in natural systems, so principally in rivers. Today, Joby's concerned about urban water systems, and this is through his excellent research in distribution pipe networks. And his research really tries to protect public health through better management of quality of supply of drinking water. Recently, Joby was head of department at Sheffield and also led the 2014 Sheffield REF submission. But today we're lucky enough for Joby to share some insights into a research topic where he really is one of the global leaders, and that is drinking water biofilms. So yeah, without further ado, over to you, Joby.
Thank you, Francis. I think I need you to stop sharing, do I?
Yeah.
Lovely. See if I can share a screen this end. Let's try for that. And that means you lot have all disappeared, so I can't see anyone. Have you got that screen okay there, Francis?
Yeah, all good.
Excellent, thank you. And thank you for that lovely introduction. I don't say much more about who I am, so I shan't. Title I've gone for today is What are biofilms doing in our drinking water pipes? Maybe challenging some people who might think that they don't exist, but probably more thinking and looking at what we've understood through a whole series of research over the last decade or so, trying to actually probe what are the biofilms like, and in terms more of how are they making that link between the water distribution infrastructure and the quality of the water we receive. So I'm going to give you when I looked at the delegate list, it looked like we had quite a range of different backgrounds. So I'm going to give you a little bit of an introduction into why do we care about water distribution systems, as well as then more of a medley through those various highlights of all sorts of different bits of research. And hopefully they'll coherently come together to give you a range of different ideas and insights into what we've managed to study. But I thought I would start with what are drinking water pipes. Unless you're a water company, and sorry, I'm going to bore some of the water company people who are joining us, you probably don't think about the pipes that supply our drinking water ever, maybe until it goes wrong. If you do, maybe your view is something like this backdrop here. This is a drain down pipe, empty pipe, a fairly new one being inspected by some kind of a crawler. So this thing in the middle with the lights is a robotic device on a tether, the red light. And you can see it's this idea of a lovely, smooth, clean, perfect cylinder, dead straight. And life would be probably fairly easy if that were true. Perhaps when someone mentions distribution, these are more like the sorts of images you come to mind. Francis mentioned we have our annual media storm about leakage and how the water company should be doing more, but obviously don't have enough funding to do so through the Ofwat regulation. Maybe think about these kind of muddy holes where repairs are being done when you see them being disrupted the traffic flow. They maybe give us a bit more of an insight into what we're really dealing with here. Because what you can start to see in those is some of the ideas of the complexity of the buried infrastructure, the age of the infrastructure, the diversity of the infrastructure. And it's really this buried, forgotten, added to piecemeal nature of it that gives us some of the interesting challenges that have kept me interested for quite a while. So really, how our drinking water supply operates, if we start in the kind of top right here, we have some kind of sources that are either above ground or sometimes they're groundwater, with varying levels of catchment management to help protect that water where that's possible. These days, very high quality and well managed treatment processes delivering very high quality drinking water. Important though is it's not a sterile product. If you were to treat water to a sterile state, you really wouldn't want to be drinking it. So this idea of some kind of perfect product from the drinking water that's not going to change through to our taps is really quite wrong, because it's not sterile. Very low levels of organics and nutrients in there these days, maybe more varied historically. And then we put it into our pipe networks, these vast systems that get that high grade product from our treatment works through to the customer's taps. And it really is huge infrastructure, which is where some of the interest starts to come into. This image on the bottom left. Thank you, Yorkshire Water, for some time ago saying I could use that image. It's the Yorkshire Water region looking at something of their pipe network. So you can start to get an appreciation of just the sheer scale and complexity of these pipe systems that convey that product to our homes. The figure from a database for England and Wales combined is if you were to put all of that pipe end on end, you get around 330,000 kilometres. So that, if you were to stick it end on end, would just about get you to the moon's orbit. So us moving water around just England and Wales is equivalent to getting water to the moon, which is quite an incredible feat. That's what we do all the time, totally taken for granted. These systems aren't designed and built, not like a building or a bridge with a specific purpose and function, because they have such a long lifetime, they very much are evolving and changing and added to and messed around with. So we get lots of uncertainty with exactly how they're connected together, certainly what condition they're in, different materials. But above all, it's old infrastructure, which brings me to this kind of picture on the top right. This is, again, from a national database that looks at the date-laid information we have for pipes. And you might think that this graph's not bad. You know, the majority of this is kind of 1950s onwards. Well, that's already 70 years and upwards and downwards age of pipes. The scary thing is, though, when you look in that database, there's about another 60% by length where we don't have a date-laid record. And that's probably mostly those older pipes, again, where through the different regulation, different business structures, we've lost those records over the years. So actually, this is probably quite a significant underestimate of the age of these pipes. And of course, they degrade, they change, they get messed around with, they corrode, etc. over that 100 years plus lifetime. So dealing with those to try and convey our better than food grade product is a real challenge. And my contention, usually at this point, is that this infrastructure, these holes in the ground, are so valuable to us for getting our water supply. We're so dependent on that. We need to find ways to keep using them into the future in the face of climate change, population growth, etc. But the challenge is, how do we make best use of that old infrastructure going forward? And really, can we better understand what's going on with them? And often what people forget is we hear lots of headlines around leakage and we think about quality, we talk about climate change. We've had a threat of droughts and hose pipe bans this summer, and that's what you hear lots around. What people often forget, though, is the reason we have this infrastructure is for the drinking water quality. And I forget where I stole this image from on the internet, so apologies for no reference and apologies to whoever I stole it from. But this is a caricature of Jon Snow, who is certainly credited with popularizing the link between human health and water. So for those of you who don't know, he was Victorian engineer. The first instance, really, a graphical information systems, GIS, he was plotting cholera outbreaks on a map and had a correlation between cholera outbreaks and certain wells in Soho in London. So what he did was to go along and remove the pump handles from those wells, so people had to walk further for their water, observed a reduction in cholera, and of course, being a good scientist, was able to then go and return the handles, which we best not talk about. But he was popularised as really making this link between water quality and public health. So the reason we have drinking water systems of the nature we do, evolved since the 1800s, is really to provide that public health benefit. The weird contradiction, though, is because we provided that so well through the infrastructure we built. The biggest single use most people have for drinking water, this incredible, better than food grade product, delivered 24/7 for a bit over £1 for most of us a day. The biggest use is we flush it down the loo. We only use about 5 litres a day actually for drinking, preparation of food. So we're actually asking pretty incredible things of this infrastructure, but we really must keep focusing on the drinking water quality above all from these systems. So that's where my kind of preoccupation comes from. And you may think, well, that's fine. You know, I don't ever get ill from drinking water. I mean, what's going on about? It's not a problem. We manage these systems. We do, and drinking water is fantastic quality. I would still much rather drink drinking water than any bottled water, not just from a cost and sustainability point of view, but also from the actual quality it achieves. But when we do explore deeper, and in the UK, our GPs, our medical services, aren't required to formally record cause of illness. So we're fairly limited in this country. One of the works I always go back to is Hunter, who's a UEA, got on the press lots for COVID stuff, but prior to that, he was also looking at public health from drinking water. I think it was the DWI commissioned a study he did, and he found some fairly scary correlations between low pressure incidents and diarrheal disease, with some fairly scary numbers associated with that. In the US, things are a little different. GPs there do associate cause where they can, and a fairly recent study was coming out with some very big numbers, you can read for yourself on the slide, around the impact that has and the cost that has associated with it. And their work, they were particularly, the quote there in the middle, caused by biofilm-associated pathogens. So they really were pointing the finger at what goes on in biofilms in our water systems and the role that has in public health, and the fact that we don't know and understand enough of that. So what people maybe are more aware of, and the things certainly the water companies and Ofwat are quite, and DWI are quite driven by, is a more aesthetic issue, gets called discoloration, although it's not true colour. It is fine particulate material. The graph on the right is from a website just called Discover Water. It collates and represents lots of the regulatory information the water companies have to submit. It's publicly accessible, so that's just a look at the numbers of customer contacts that the different water companies receive. And it is an unpleasant experience if you're unlucky enough to get it coming out of your tap. Fortunately, it's often quite sporadic. It's short-lived events, goes away, and it's generally caused by hydraulics disturbing material accumulated within pipes. So at Sheffield, we're quite known for our discoloration management research, helping the water companies really improve and make quite dramatic improvements over the last decades in their discoloration. But it's still considered unacceptably high levels. Worryingly, and the DWI are on record as quoting it's a mask for other water quality issues. When you've got discoloured water, there is often a lot of microbial as well as inorganic material in there. So it becomes a very interesting driver and a topic of why we care, why we're interested in what goes on the inside of our pipes. So that's really what I've been obsessed around for quite a long time now, and trying to understand is the fact that we don't have these perfect, smooth, clean cylinders magically conveying this inert substance from our treatment works through to our taps. What we actually have is these huge systems, really high surface area, complex mix of materials, changing over time. So these top 3 images here are bits of cast iron pipe. So you can see, as you'd expect, kind of corrosion deposits inside some of them. This one's a more interesting one where you can see really big accumulations of material. Yes, some loss, some graphitization of the wall, but this amount of material isn't just loss of the corrosion of the cast iron itself. This is, again, these accumulations from hundreds of years of very low background levels in the water and how it can accumulate. And the top right one here, this is a section of plastic pipe. You can see the kind of blue plastic material where it's been cut through. And again, you can see the slimy, in this case, dried out slime. And our discoloration work usually goes under prediction of discoloration in distribution systems, which has been smeared inside this pipe by one of the postdocs who was working on it. But again, you can see this slime, this idea it's stuck around the entire inside of the pipe. So lots of our research is that discoloration isn't, although it's particulate, its behaviour is not driven by the kind of self-weight sediment. It's not an invert deposit. It's these slimes all around the inside of the pipe. So the bottom right one is a real favourite image I like. This was a large diameter trunk main, 1.8 metres diameter, was taken out of service for various reasons, including discoloration. So big that the engineer could walk inside it, and you can see he's bravely smeared his hand around the inside of the pipe, and you can see the iron and the manganese rich, so the oranges, reds and the blacks. And it is the inorganics you tend to see, but you can also see it's formed these really slimy, sticky, cohesive deposits, which really starts to, probably the people on this call, make connections with biofilm behaviour. So the reason I get kind of obsessed with the surface, not the bulk water, is again, if you go through and draw things like that database and you work out areas and volumes and pipes, you get to this kind of interesting headline that for every metre cubed of water we've got going through our systems to our taps, there's 11 square metres of pipe surface. So yes, there are changes in the bulk water and they're important, and we have a good understanding of those from our treatment processes. It's actually what's going on those surfaces that are dominant and where we have much less certainty of what on earth is going on. So I showed you an image earlier of kind of a hydrant being opened and dislodging some material. That's a very common way for us to try and look and understand what material is built up inside pipes. Because they're buried, because they're underground, they're under our roads, actually digging them up and accessing the pipe is a real challenge. So the easiest way is to open a fire hydrant, create some more aggressive hydraulic forces in the pipes normally useful, and observe the material you observe, that you immobilize. Very common kind of cleaning strategy the water companies use. If you collect samples kind of every minute, every 2 minutes through flushing a long pipe, you can see this progression of initially really dirty material and slowly improving as you mobilize through as more cohesive layers. And you can see the turbidity, a measure of how cloudy the water is, the iron and the manganese, and a cocktail of all sorts of metals, and indeed that it is particulate rather than true colour in the water. And this is kind of how we understand the inorganics and the aesthetic reaction that's going on. The problem is if you clean it, and whilst a lot of those deposits are historic, this stuff comes back remarkably quickly. So this is an example of an extensive cleaning programme, one of the UK cities. The water company got permission and ran a £70 million cleaning programme. So they really aggressively went into all these pipes, replacing non-preferred material, lining it, cleaning everything, putting foam swabs through, all sorts of things. Thought they had this beautiful, shiny, pristine system. Just 12 months later, though, they were starting to get discoloration contacts again. And this is a large section of pipe that you can see they were accessed 12 months later. And again, you can see inside the pipe material built up all over the internal surface. And again, someone's bravely rubbed their hands on it, and you can see the iron, the manganese rich deposits that are built up in just 12 months since cleaning. So it is very much this ongoing process. So all of this, and I'm sure for this audience, you're already making this connection. Sometime back we proposed that actually, if we couldn't describe this with kind of physical processes, chemistry that we were looking into and understanding it for the iron, the manganese didn't explain it all. So we proposed that really what we didn't understand about biofilms and the fact that it's on the complete surface where all the biofilms are going to like to grow, the biofilms were really this missing link between the water quality and the pipe infrastructure. But really recognizing that the pipe environment is quite uniquely complex. Yes, it's low nutrients, low loads from the treatment works, but we have a very complex hydraulic regime, some quite high shear stresses, complex turbulence processes. We have complex nutrient gradients, we add chlorine residuals, and we add all that on top of the normal kind of complexities of biofilm growth. So we set about, could we understand biofilms better to help us particularly understand and manage discoloration? So you'll see lots of turbidity data through the plots I'm going to show you. So that's rather a long background and context. The next question is really, how do we study these? I've said it's really hard to access buried pipes, to take samples of the biofilms and have a look at them. Yet the pipe environment is really complex and unique. So what we built at Sheffield over the years and run is a very unique international facility. So this is comprised of notionally 4-inch, but it's 80 mm internal diameter MDPE or HDPE pipe rather. Very common what the water companies put in the ground now. It's coiled up in our lab, although it's still a big room full. And what we can do is run our standard lab water as a base condition, put different hydraulics, different temperatures, dosing of different substances, which I'll show you some results of later. We then run different hydraulic regimes in the 3 loops. And importantly, we decouple the hydraulics of the loop versus trickle, renew and turnover of the water through each system with its own pump and tank system. So we can really study and recreate accurately the full pipe complexity of conditions we're interested in. And then importantly, we put some coupons into the pipe wall. So when we drop the flow and the pressure, we can access these coupons from different points all along and around different bits of the pipe. And we can take them out. That enables us to really study the biofilms that have grown on the pipe. Importantly, we make our coupon sections out of pipe material, so they actually have a slightly curved surface, so they do fit flush or reasonably flush to the internal surface of the pipe. So we maintain all those boundary interactions, the nutrient exchange, the hydraulic shear stresses that are imposed on the biofilms we grow. Coupons really important, and I'm going to show you lots of different results of the different analysis we've done with these coupons over the years. We tend to split them into 2 with this inner bit that does have a genuinely flat surface, so we can put that under a microscope and not worry about the curvature. So we look at both the biofilm in all sorts of different ways, so kind of the community. We're doing sequencing work to see interesting stuff about who is there, We often these days tend to use flow cytometry rather than some of the more complex staining techniques with some of the early data I'm going to show you. Great for giving us a measure of total and intact cell counts that we've got on them. Again, from that outer insert, we can often go and do some fancy X-ray diffraction so we can get the inorganics that are bound in their information. And I'm going to show you, we've been looking a lot at kind of the physical structure. So how is it getting strength and bound to the wall? So that's often trying to characterize the EPS, the extracellular polymeric substance. So usually the carbohydrates and the proteins. We're dealing with really small levels of biofilm compared to lots of other environments. And so the kind of extraction-based techniques really don't work for us. So we have developed some techniques, and I'll show you using confocal laser scanning microscopes and triple staining. If anyone through the course of this talk is aware of other quicker ways that we can quantify small levels of biofilm, even if it's an overall measure of cells and other material, but particularly if it's ways we can quantify the different characteristics of the EPS, I'd be really excited to talk to you. But I'll show you some data what we're up to first. So these are just some SEM to give you some ideas of what we're dealing with, what our biofilms look like. This is our clean HDPE, high density polyethylene pipe. It's an extruded material, it's a super smooth, shiny material, so it's lovely in terms of low head loss, so great for transporting water a long way with minimal head loss. When you look at it close enough under an electron microscope, you can see it isn't perfectly smooth. You can start to see some of the ridges and the forms from where it's extruded. You still get some scales and things, but it is still fairly smooth by most surfaces standards. If after, say 28 days exposure to our flowing conditions, this is more like the sorts of images we get. So you can start to see even after 28 days, we've got some nice biofilm developing within our drinking water environment. So you can see, if you zoom in again, some kind of primary cell attachment, so you can see the cells and the beginning of some EPS sort of frondy bits, how they're sticking and attaching to that super smooth plastic surface. There are some areas of much more developed biofilm. So those of you who work biofilms, that's probably much more sorts of images you're used to seeing, these sort of spongy or kelpy seaweed kind of structures, but a much smaller scale, as well as all these channels through it where material can get trapped and do all sorts of interesting things. As well as the biofilm itself, you can see inorganics incorporation. I think it's actually clearer here. This blob here, you can see something much more kind of crystalline and inorganic stuck already within the biofilm. There are some more bits of it here in this more developed biofilm. So we start to get to our more kind of traditional, I guess, view of biofilm of, yes, it's the cells, but it's also a lot to do with that EPS that the biofilm produces, helping it to stick to the wall that can then incorporate the inorganics, channels and then voids within it, and a really complex, interesting environment. Although it's really, really thin, and you can see from the kind of zoom level these images, we're talking tens, maybe up to hundreds of microns of material stuck on the pipe wall. But again, over that vast pipe surface, that can have a really big dominant impact on our water quality. So this is a kind of a method for lots of the results you're going to see all in one slide. So typically we run our pipe loop with at least 3 different conditions in our 3 loops and then repeats to get other variables. We tend to run it for a month at least. Some of the results will be for longer, but typically a month is long enough to get us some biofilm developed and measurable response. Then when after a month, we simulate a hydraulic disturbance. So we increase above the sort of highest it's seen over the month and put some stepped increases of flow and observe how the biofilm is mobilized into the bulk water, as well as then by taking coupons afterwards, what's left on the pipe wall. So a typical result, the first one I'm going to show you, is looking at the impact of hydraulic conditions. So we might look at a low steady state flow, a medium and a high steady state flow, and we can look at just the effects of different rates of exchange, different shear stress regimes going through the pipe. Our systems, our pipes and our systems don't actually experience steady state flow, though. You and I all use water with quite a defined diurnal pattern. So what we usually observe is a peak in the morning when we all get up, shower, etc., a lull through the middle of the day, then a longer, flatter peak in the evening, and then a real low through the nighttime. So you're seeing 2 kind of typical diurnal patterns that we might see in operating systems. These have been quite contrived to fit in here so that they're bracketed by the high and the low steady state, and the net 24-hour flow is the same as this medium flow rate. So we've got these three kind of hydraulic conditions that hopefully you've had a look at, and we can look at how just that hydraulic condition impacts the biofilm we grow and who is there, etc. So our first set of some results for you to look at. So a snapshot here of those three hydraulic conditions, so in black and white on this slide. And I'm going to skip the complexity, the bottom right set. I'm going to draw your attention to this top leftish slide. This was Dappy staining under a scanning microscope, just to look at the amounts of cellular material. And you're looking at kind of the darker greys after the 28 days, and then the end of flushing. So after we've mobilized material, so you can see the really low steady state flow rate gives us most material and most mobilization, which is exactly what we kind of expect. The medium flow rate gives us a bit less mobilization maybe. The high flow rate, something a little bit odd going on, that we had more material after we flushed on average, but again, less material than the other two. What's really interesting here is when you look at these two varied flow rates, they have less material again. So we get this idea, and our concept of this is that overnight time, when we've got this lower flow, we're getting enhanced biofilm growth, material accumulation on the wall. But every morning we're getting that stripped off by this morning peak and then moved away, transported through the system. So the next night starts to build up weekly again, then is stripped away. So this idea of a kind of daily cycling of the biofilm growth and mobilization. And it's definitely the biofilm because this is the measure of cellular material with a Dappy stain. Interesting result of why the low varied is less than high varied flow, which I'm hopefully going to explain on a later slide, but we didn't know when we did this experiment, which is quite a while ago. As well as looking at kind of just the amounts of who's there in the discoloration response, we can do some gene sequencing work. We can have a look at who is there. So when we first started doing this, we thought, well, can we associate a certain community with the less well-held material? Are there certain parts of the microbial community, the bacterial community, that are more associated with the biofilms and more associated with the biofilms that are mobilized by flushing? So the stacked bar charts, I know they're a horrible way to present data, but the left of this split, if you can see my laser pointer, is the biofilm data. The right is the water. So if you look at the biofilm first, again, we're split into two here, and we can see the kind of before we flushed, so after 28 days of growth, then after we flushed them. And it's kind of disappointing for the theory we put forward, or the hypothesis. We don't actually see a very pronounced difference between who is there after 28 days and who is there after we'd flushed. That there's less material, we've seen that in the previous slide, but it isn't any particular part of the community that is more associated with the weaker layers, the more readily mobilized material. There's pretty much the same kind of community, give or take, between the before and after. The thing that really dominates on this slide, and maybe is most clear in these MDS plots, is there is a really defined difference, the different levels you look at here between the bulk water in our system and what grows into the biofilm. And actually, as we start to look at different waters and different conditions, we find that the biofilms have quite a common community. Yes, there's variations as a function of the water quality, but they are always distinct and different, or a subset of the bulk water. And there's quite a high degree of similarity between the biofilms, even between quite different source waters, bulk waters going through the different pipes. So kind of disappointing that who was there didn't really help us massively manage, or targeting a subset of the community wouldn't help us manage discoloration and the biofilm risks. Substrate, though, is important. This is now a quick look at diverging into field work. Our pipe rig is all plastic, so it's pretty hard for us to look at wholesale pipe effects. And you can guess which water company we were collaborating with here. And I also acknowledge all the water companies have given us great support with field sites, field work over the years. So thank you to all the water companies and all the individuals who've helped us over the years. This one, as you might guess from where that yellow blob is United Utilities. And when they gave us access to a DMA, we actually went back to this one a number of times. The result I've picked out for you here is flushing and looking at what you mobilise from a cast iron pipe length right next to and fed off a plastic pipe length. So we're looking at what was different in terms of the who is there. In what's mobilized, we couldn't look at the biofilm that was left stuck after flushing, because it's the buried pipe length. But what you can see is again, it's clearest in these MDS plots. The blue blobs are different time points of the plastic, class and species level. The browny, blacky blobs are from the cast iron pipe. So again, you can see there was some difference as a function of the subset of the material. So there is definitely some evidence, and we found more kind of iron and manganese sequestering bacteria in the iron pipe. So we're definitely more prone to using the substrate material as a source of iron itself, and more of them proliferated and managed to colonize over there in the cast iron pipes over the years of its service. So the substrate material is important. That's my one foray of it into this talk. Everything else you're going to see is from the plastic pipe loop. But substrate does have an important role. So having not got the kind of insight and answer we were looking for from purely who is there, having understood more of the role of EPS, I mentioned we developed a technique to try and understand the EPS in our very small biofilm layers. We tried most extraction techniques, just not sensitive enough. So what a very good PhD student now, postdoc with me, Catherine Fish, developed and applied was a triple staining technique under a confocal. The data you get is absolutely fantastic. The time and effort required is absolutely prohibitive. I would love to go back and use this technique more. Very hard to justify the time and the effort. So again, if anyone has ideas of better, faster ways this could be done now, be delighted to hear from you. What CAP did was to identify 3 different stains that we could put on that had different excitation emission spectra, distinct again from the autofluorescence of the plastic underneath itself, that we could get a measure that was more associated with cells, carbohydrates and proteins. Then I was under a microscope in a dark room to image little areas of our coupon inserts that have a flat surface that can be used under the confocal without distortion by the curvature. Then unmixing them and quantifying each one, very, very time consuming, fantastic data. So the sorts of things you can get from that are 2D stack images for nice quantification and area coverage. Or if you have colleagues who like to do things in aerospace departments, they can even do these funky kind of fly-through images of one of the fields of view. So here again, you can nicely see, I talked about it being, if I can get that to run again. I'll go back a slide. Oh, here we go. It's an extruded surface, so it tends to have scrapes and grooves in it at a microscope level. So again, you can see where the biofilm is preferentially liking to start growing and attaching to those grooves in terms of its structure. And you can see the different cells, carbohydrates and proteins in the different color. Not sure what the value is, but those fly-throughs are fascinating, but quantifying the different amounts does give us some good insight. So I'm going to take you back to, again, the steady state, the low varied and the high varied flow. We had that anomalous result that the low varied flow gave us least material. Actually, what we found when we looked at the who's there in some detail, the steady state community was a little bit different to the low varied and the high varied flow. So that selective pressure of the cycling of the hydraulics that appeared to give us some benefits in terms of managing the amounts of material and discoloration risk was also giving us a slightly different community. Drill down to just look at that level. When we then looked at the cells in blue, the carbohydrates and the protein, again, they're a little bit different across these conditions. But actually, the reason we were getting least discoloration risk from the low varied flow is that had by far the highest ratio of that non-cellular material. So that the carbohydrates and the protein combined, that was in turn giving strength to the biofilm, letting it attach well to the wall. So again, it said the pressure of who's there, but then how who is there is producing their EPS matrix, how that is then enabling to protect themselves attached to the wall. With the caveat, this is a 28-day experiment. This is probably quite a young biofilm, as in most of these results that we're looking at here. But again, decided we need to look much more than just who is there to understand the emergent behavior we're interested in, or I'm interested in. So another interesting one, important one to look at is temperature effects. Naturally, our distribution systems vary over the year. And maybe we're interested in the effects of hotter climates as we head towards climate change scenarios of the future. So the first set of results I've got for you here is more kind of looking at winter, summer we've got at the moment, but also as a cross correlation of that with hydraulics. So I'm sorry, this is a really complicated slide to get your head around. These first two plots on the left are those same 3 steady state flow rates at 80 degrees at the top and 16 at the bottom. The next pair are the low varied, the steady state, and the high varied flow, again at lower and higher temperatures. Be careful that the Y scale range is different for the 2 hydraulic conditions. But if you compare 8 degrees and 16 for the steady state, you can see at the 16 degrees there was more material being mobilized. Again, the 8 versus the 16, you can see there was more material for different varied flow patterns at the higher temperature. And again, you can see the result we've seen before, counting for the error bars, more material at the steady state flow rates. And again, if we look at the cell counts for these with a bit of staining work, you can again see the colder temperatures had less material after 28 days. The higher temperatures very much had enhanced cellular material correlating with this enhanced turbidity that we see at the higher temperatures. So very much a cross correlation of the temperature and the hydraulic effects interacting with the selective pressures, both combining to influence the amount of biofilms and EPS, and therefore the more visible metals that we're trapping and then mobilizing to give us the turbidity effects. Maybe though, it's interesting to also look at how temperature affects who is there. This is a snapshot looking at the who is there, but for going for higher temperatures. So I'm going to actually point you to this slide in the middle top, showing the colder and the hotter turbidity. Again, higher temperature, we saw more bacterial growth, which is really clear in these SEM images. So the top pair are 2 zoom levels at 16. The bottom pair are the 24 degrees at same 2 levels of zoom. And you can visually see much more material at the higher temperature, giving this higher turbidity. The bacterial community, it is a little bit different. And we've seen these stacked bar plots at the 2 temperatures. Interestingly, and I've not mentioned it from the data so far, when we look beyond the bacterial and also look at the fungal community, we are often finding a really core fungal community that doesn't change too much. But going to this higher temperature really did give us quite a change in the fungal community. So the fungal as well as the bacterial communities are playing a really important role in what's going on and how these layers are forming and growing inside our pipes. And when we went to this 24 degrees, there was really quite a marked shift in the fungal community we've got growing inside. And those of you with sharp eyes, I think you can probably see more sort of fungal spore shapes within these higher SEM images than you can see in the colder 16 degrees water temperature. Excuse me a second. So another interesting bit to look at, and lots of time now, I better speed up, is to look at nutrient levels. And one of the key ones in drinking water that we think is the limiting factor is the assimilable organic carbon content in the water. Again, very good research. Fran Pick developed an improved technique to look at measuring AOC that combined it with the use of flow cytometry. So this is top plot is showing kind of the development work of that. The bottom plot is kind of raw and treated water, giving you an idea of how varied AOC is across, I think that was a dozen, maybe it was 20 different treatment works with a collaborating water company. Thank you. I'm sure the company knows who it is. Thank you for that support there. This is a work then looking at how that AOC changed through networks and the inlet and outlet of service reservoirs. It's kind of complex to see in there, and I'm going to rush through it, sorry. What we observed was that when you had purely a change through the service reservoir where bulk water dominates, we saw AOC being consumed. Where we had a pre and post the end of a pipe length, generally, and it was pipe dominated, therefore biofilm dominated, we were tending to see AOC actually produced in some places. So a hint of really complex AOC cycling within and through the biofilm. So what we then went on to do with Fran was to build some mini pipe loops and put them at the water company's treatment works, and basically picked 3 sites that were fairly dominated by lower, medium and higher AOC. Again, you can see in the SCM images, this was actually run for a year. So this is much more mature biofilm than most of the other data I've shown you and plots I've shown you. You can really clearly see how different the biofilms that we grew inside the pipes of the system were. Although AOC was a dominant difference in these systems, it's not the only one. So you can see cell loading was quite different, as well as a few other factors that are in the table there. But AOC was an important factor in the difference of those. And what we went on to do with Fran was to monitor how the biofilm grew and then was mobilized. So these are cell counts total and intact from flow cytometry. And you can see a nice growth curve for the high AOC that was actually starting maybe to flatten over, then the medium and then the low. And you see the kind of the food source, the nutrient source, the AOC having a really dominant effect in the amount that was built up in those biofilms over the year. These four at the bottom, the top two and the bottom left, are all showing turbidity, total organics, and iron, kind of a gradient of increase as a function of those different AOC conditions. Interesting, when we looked at the AOC released when we flushed these systems, they were started at different levels, but actually released the same amounts. Of the gradient of these three lines are much more the same. So the AOC actually incorporated within the AOC was fairly constant, which again supported our idea of kind of much more complex AOC cycling by the biofilms. And actually there's this nice image we drew to try and then capture the ideas we got from this data and the previous data of the AOC cycling by the biofilm, but the biofilm not actually acting as a store of AOC within the pipes themselves. And I'm going to actually skip that one because I'm running out of time, and I want to talk to you about this data. So one of the other important environmental conditions that we manage and look after is the chlorine residual. So we add a chlorine residual to our treated water as it goes into our networks, with the idea of trying to limit regrowth in the bulk water, but also protecting against contamination. So what we did in this case was to put our normal Sheffield tap water in with our residence time getting us about 0.5 milligram per litre. In one loop, we then dechlorinated, dropped the chlorine down. In another loop, we were boosting it up. So again, most of the time over the month, we had that control. You can see a few periods in time, we lost it a little bit. With the idea that if we had higher chlorine, we'd suppress microbial cells in the bulk water and probably the biofilm, and we'd have less discoloration risk. We actually found quite the opposite. So the first time we ran this, we assumed we must have done something wrong, mixed data up, goodness knows, couldn't find any errors. So we then repeated the whole experiment. And again, you can see the high chlorine regime is the kind of yellowy data, the highest turbidity. We actually repeated that for a month's growth from a clean system. And the second set of data here is after we'd flushed it, we then let it grow for another month. And again, you still saw the highest turbidity, iron and manganese, from that high chlorine regime. Totally the opposite of what we were expecting. The cellular response, so this is again flow cytometry data, total and intact cells. As you'd expect, most cells where we had the dechlorinated regime, least cells where we had the chlorine boost, and we kind of confirmed that with some qPCR data, which again supported for the bacteria. Interestingly, again, this hint that fungal community is important. The fungal response was much more independent of those different chlorine regimes, so having an important role. But again, the real insight into what's going on here was how the EPS was being produced under these different regimes and giving you the different responses as a function of it. So really the key was the selective pressure of the chlorine influencing, yes, who is there a little bit, but much more how they produced EPS to protect themselves, therefore what that EPS enabled the community to evolve to, incorporating iron manganese or not, or staying attached to the pipe wall. So really this result showed that chlorine had the opposite effect of what we were intending. Of itself, though, that is not a message to not add chlorine. It does a great job in protecting our bulk water, but maybe very high levels of chlorine are not necessarily something we want to go to, and they're probably not something we want to go to for the risks of disinfection byproducts formation as well. So just about keeping to time, a bit over. Going back to my posed question that I gave as a title, what are biofilms doing? Well, lots, and there's lots of them. I think it's a really important message. But also, although I've spent a decade or more trying to understand them and really only scratching the surface. We need to understand them much more. It's really important we look beyond simply kind of who is there from the DNA fragment level. EPS is definitely really important, as is fungal. But again, just presence, I don't think is enough. Really interested in getting more of the kind of who is there, but viability and maybe even to function some of the proteomics work and ideas of applying some of those techniques that are emerging. Really important message, though, everything we do in our pipe networks, in terms of the hydraulics, the chlorine regimes, the temperatures, they all impact our biofilms, which in turn then impacts onto our water quality. So understanding those coupling effects, really important. Probably my real big takeaway that I'll leave you with, though, is that we're not going to get rid of biofilms. No matter what we do in any environment to try and get rid of biofilms, generally we create nastier niches that tend to create biofilms with probably less desirable emergent properties. So really it isn't a question of how do we eradicate and get rid of them. It's how do we learn to understand them, how do we manage them so that we promote the beneficial properties and suppress the non-beneficial ones. Very quick last plug for what we're doing next. I started talking about and mentioned pathogens, and chlorine does a great job in protecting us in the bulk water when and if pathogens can enter our biofilm systems. And we've done some work looking at the risk under very dynamic hydraulic conditions where we might get leakage or ingress back through leakage apertures we know are rife in our systems. Really interesting questions. If we are doing things like promoting protective biofilms through our chlorine regime, might they be preventing environments where pathogens might colonize our biofilms, might even grow and replicate, and then when they mobilize, when might they be a risk to public health? So really trying to look at how different chlorine regimes are impacting biofilms and their potential as this pathogen growth, proliferation, mobilization. And I'm delighted that we just had announced this summer a very large grant funded between Sheffield and Glasgow. And thank you for all the many water company regulators that supported that, and particularly John Fowle, who's going to take the brave role of trying to manage the steering group, or at least chair the steering group for me and keep us all on track. But really trying to get to kind of biofilms and this missing link between protecting water quality and our aging infrastructure, and therefore what should our disinfection strategies be going forward. Not necessarily saying we shouldn't chlorinate, although some countries do go for that strategy, but more what is the right chlorination strategy to give us the benefit in the bulk water and minimise the risks from biofilms or disinfection byproduct formation. My final slide is a thank you. So this is our rogues gallery of all the different researchers active in the drinking water research group at Sheffield at the moment. The circles are key people for the various bits of research that I've highlighted to you, and a special guest appearance from Rebecca Sharp at the bottom, no longer part of the group. She's a lecturer at Sheffield Hallam. Some of the early data I showed was from her research. And of course, a huge thank you to the various funders, whether that's research councils and particularly the water companies who've supported us over the years with funding, but also with site access, insight, steer direction for all this work. And with that, I'm probably have used most of the hour, hopefully a few minutes for some key questions, and I'm happy to stay for anyone who wishes to stay over the hour. And I'm going to try and stop sharing so I can see who's there and who's around.
Great, thanks very much, Joby. Fascinating talk. We've had a number of questions, so we're definitely going to go over the allotted time. So yeah, just to reiterate, if anyone does need to leave and the conversation does drift beyond the hour, then please, do feel free to leave. The full meeting, including the questions, will be recorded and disseminated through the EBNet website. So I'm just going to pick on some of the most popular questions, or the ones that I think are very pertinent. So we've had one around your really neat statistic of the meter squares of biofilm of pipe work compared to the water phase, and Sonia Heaven from Southampton University has asked if there's any sort of understanding around flux. So if you're talking about surface area of biofilm with a certain concentration of bacteria, how does that relate to the mass load of bacteria coming through the water?
I haven't done the flux for bacteria, no. We've looked at the flux, or tried to look at the flux for iron, So people have had ideas of ion loading into the system, and could we do net max balance to work out where we're getting loss or gain of iron through systems. The usual problem is we just don't have enough iron sample data coming out the bottom end of the system. And pretty certain it's going to be the same story for trying to do a cellular flux. People have started to apply flow cytometry quite extensively at treatment works, some at service res. The amount of data looking at the kind of downstream tap cell count is tiny. So you could try and do some fluxes, but I think you'd be kind of guessing in the dark at the moment whether we're getting net accumulation or loss to and from the biofilms. But it would be a great data set to start to collect.
Fabulous. Thanks, Joby. Something I've been quite interested in as well. There's been a question around how might we coat pipes. This is Arjun Venkatesh from Cranfield. How might we coat pipes to, prevent the biofilm from growing? So rather than trying to manage it with a disinfectant residual, could you do it in another way?
Yeah, and there are a number of products out there. Interesting, we've done some work looking at a company, which I probably better not mention, that provide anti-fouling coating for boats. And one of the interesting observations they found that drew analogies with us was when a boat sits in harbour, so it's got very static flow, you get really thick biofilm no matter what the coating. If you've got a boat that's working hard, zooming about, and then sits only for short periods, that cycling of high shear stress, then growth, is what suppresses biofouling of boats quite effectively. The problem with lots of those coatings and going on into pipes is lots of them are to a level sacrificial or something in the coating, particularly the ones that are used on boats, where they're getting better if they don't release to the marine environment. The thought of putting those near your drinking water is a big no-no, and rightly the materials approval to get that through is very strict. There are some emerging coatings that show promise and definitely don't have a sacrificial component of what would go on. The big problem I foresee with them is, yes, we could use them, and certainly putting them on new material would be great. The worry is our pipe renewal rate is well below 0.5% per year by length of the pipe. Even in bits of the country we're investing well. So the time it would take to apply that and have that right through our distribution systems is hundreds and hundreds of years. And even if we were to go for draining down pipes, clean them well enough to spray or apply some coating that didn't have to be applied in the factory, it's still going to take us a very, very long time to do that. So yeah, there's a place for it, but equally we've got to still work with the online pipes going forward as well.
Fabulous. And then one from John Fowle, who's at Surrey and also Cranfield as a visiting professor. So mentioned about different materials and the role that plays. I guess you did go into that in the talk, so perhaps maybe we can focus on that later. We've had a couple more here around the presence of algae. Have you ever detected sort of microalgae growing within these systems? Not looked for it.
Not going to look for it, but there is definitely some interest in kind of getting into archaea, and particularly with Legionella is obviously a pathogen of real concern, very associated with archaea. Not particularly gone to try and look into algae though. No, it would be an interesting one to look at. Obviously, get the algal blooms in certain surface waters. I think most treatment trains that we've got these days are pretty effective at taking them out, but there's still some carry through, whether they're managing to proliferate in the environment in pipes without the light. Not sure. Never looked for it.
No worries, thank you. And then there's one from Louise Byfield, who said, are there different sort of safe and pathogenic biofilms depending on conditions, or is it just about the quantity and therefore that impacting the health risk of water?
Yeah, really good question. I think the answer is honestly don't know, but that's what we're really keen to find out within this next phase of research we've got funded. Where we've gone looking for pathogens, yeah, you can find certain DNA fragments, but often in lots of environments, if you go looking hard enough and you search for fragments of DNA, you'll find all sorts of things that you can associate with things associated with pathogenic strains or not. Whether they're viable, though, is really a key question. This is why that kind of last hint of getting beyond just finding certain DNA fragments and looking at the who's there from that sense, but actually coupling more of the viability with who's there. So I think I said at the start, I'd still very, very happily drink tap water. certainly preferentially over bottled water, I still think the health risks are tiny, usually because when we got the biofilm breaking off, you see it. I'm unfortunate enough to live in a region where we do get discoloration problems. You see it, you know that's not water you should be drinking. Flush your tap till it's gone.
Great. Take home message: drink tap water over bottled water. Absolutely. And then one final one, and I appreciate we're now over time, so if anybody does need to leave for the next meeting, please do go ahead. Again, this is from Sonia, who's suggested that this presentation has described some really interesting, insightful ways of which environmental and sort of engineering aspects might cross over and affect the biofilm. And there's obviously this desire to reduce discoloration, other negative effects of biofilms. She's asked, is there a value in hosting a discussion on the combined impact of these factors to try to help manage biofilms better? So basically, is there a secondary discussion that needs to be had? And also, Joby, do you have a research paper that you might be able to share with the group afterwards on this topic?
Yeah, I mean, usually most of my research interest sparks up at the outlet from the treatment works, but there's a huge coupling from the treatment works through to what goes on inside the pipes, and even upstream from the source and how the source is managed, whether that's an upland catchment or groundwater. Then what the treatment train does, what standards you try to achieve through there. I think it was Amit Pinto was looking at communities on filters and then communities in the bulk water in distribution networks, and was delighted with the finding. I think the number was something like 40% similarity of filter community and bulk water community. Basic question, well, what the hell is the other 60%? Where does that come from? Which I think probably the biofilms in the pipe and the legacy, but definitely coupling more, making sure we don't try and draw this hard boundary between treatment processes and distribution systems is really important. There's lots more we need to discuss there, both in terms of what the treatment works lets pass through in terms of the community, and it can't remove all of them, but also in terms of the different nutrients that we're putting into the water or passing through, and the background levels of iron, if that's a coagulant, or from the source water, levels of manganese coming through. Lots of the biofilms aren't all that visible. So if we could cut down the iron, the manganese, which is the main bit we see, lots of that discoloration risk could go away. But usually in terms of getting the iron, the manganese out, we're also getting the nutrients down to really low levels as well. So yeah, definitely lots of place for that discussion. Brilliant.
Okay, fabulous. Thank you. Yeah, Sonia, if there's any aspects that you'd like covered as well, it's quite a long question there, so I might have missed some aspects. Do feel free to get in touch and contact Joby directly. Any questions from the floor actually on that point? We haven't opened up to the group, so if you do want to ask a question, please switch on your camera and your mic and go ahead.
Hi, Joby. I have a question about bacteriophages. What's your view on using bacteriophages to control these biofilms?
Mixed, mainly because ultimately you're putting it into a substance we're all going to drink and consume in quantity, and the uncertainty of what they're then going to do in you and I think probably prohibits them most of the time from my concern, unless you're going to do a system where you are somehow isolating a part of the system from any consumers. You can use them in a dosed response and be certain you totally remove them from the system. I struggle with how we would want to sanction their use and their mass exposure to individuals unless you're very, very sure what they were doing and impacts on the human health downstream.
Perfect, thank you. Phage therapy is always a controversial topic, but one that's worthy of discussion. Joby, one from me. Do you feel that there's value in us ever distributing sub-potable water up to the point of people's homes, and then having dedicated drinking water taps or possibly showerhead disinfection systems to sort of decouple the distribution and treatment of water and try and manage water slightly cleverer?
I'm totally distracted by seeing John Fowle put his camera on, shaking his head.
I just want to agitate John.
So
really, I'm here.
Clearly.
That's my job role at Cranfield, to agitate John Fowle. So,
it's a really interesting idea to play with, and particularly that statistic that, you know, of the 150 litres per person per head we use on average, that only kind of 5 to 10 of it is really for those potable purposes. So why the hell do we treat all the rest of it to those potable standards? Lots of the problem of that is though really in the social aspects of our behaviour, our practices around water. And then how do you, particularly maybe in individual homes, if you have no visitors, you could do it, and certain homes do. I mean, some have private supplies, they have the membrane, the UV under the sink. And I was quite perturbed to have rented a holiday cottage a couple of years ago, and after a few days there to discover that's what we were drinking. And I didn't know how well they'd been looking after their filter and their UV system. And I think that's the big concern. Who then becomes responsible? What happens when you have visitors? What if that is in a much more public building? How's it then looked after and regulated? How do we control behaviours around it? So from a purely technical point of view, yeah, it makes lots of sense. But why would we treat all that water when we don't use it for that purpose? It's how you manage, regulate, control, and look after behaviours that I think lots of the real challenges for that kick in. But technically, yeah, it's a nice idea.
Well, I think it's being put into practice in Saudi with that new city, because they're going to distribute separate mineral water that's pre-chilled without chlorine, and then have a separate supply, which is basically potable grade, but sort of slightly less energy put into it, I guess. So that dual distribution.
Yeah. And I guess if you're, a lot of the risks depend on how non-potable your second grade is. But even where it's you know, certain cities and places have the purple pipes for irrigation water, it's a much lower grade. There are still there, even when those don't get into the home, fears of a kind of unintentional cross connection of complex plumbing systems.
Perfect. Lovely. Thank you very much. So any further questions from the floor? John, I can see you're itching to ask a question as usual.
I think we haven't got time, and it's just, I'd like to say thank you, Joby, because it's such a fascinating topic, and it's an important topic as well. And there are so many different areas that you picked up on, and the questioners have picked up on as well, which have been really good. And that last one is interesting. And you picked up on one thing, is energy, the energy use in distributing water. So one of the things I would like us to look at is the impact of smaller pipes on biofilm growth.
Yeah, yeah, definitely be a thing to look at. I think energy and chemicals, and one of the drivers around the right chlorine regime is better justifying the levels of chemicals we're using. So a bit of work I didn't get to touch on is the role of phosphate. We obviously add also phosphate to help manage our plumbo solvency for the bits of lead pipe we've still got knocking around, which is sadly still too much. Phosphate's obviously an interesting food source. So we've done some interesting research looking at how that impacts. It's not often the limiting nutrient in most of our systems is what we found. But there are some bits of certain companies' systems where they're confident they're 100% lead free and starting to switch off phosphate, which is having some impacts. So yeah, chemical energy uses definitely need to better understand. And thank you, John.
So the chat is carrying on. So I mean, clearly this has been an excellent presentation. So yeah, on behalf of everyone, thank you, Joby. Yeah, if you do have any further questions, please do direct them either directly to Joby or through myself or the EBNet admin team, who'll be happy to sort of coordinate that discussion. So with that, I'd like to close today's seminar. With that, thank you very much.
Excellent.
Thanks all.
Cheers.